First of all, I would like to thank the organizers, National Academy of Medicine, for the invitation to lead this session. So my name is Tom Cheung, and I'm from the Hong Kong University of Science and Technology. So I think I'll give a very quick introduction about myself. I spent approximately half of my life in Hong Kong and the other half of my life in the US. So basically I did my training, undergrad all the way to a PhD postdocs all in the US. And then I came back in 2013 to Hong Kong, where I grew up, to start my lab. So, I would say it is very interesting ride, and it is something that I truly enjoy, being a US citizen in a different place and learning different thing in different countries. So currently I am helping the university to run a biosciences central facility, which has about 70 equipment. And that includes different kinds of proteomics, NGS, flow cytometry, and imaging core. So basically, we have a full-blown, full-service facility, and everyone at the university and in Hong Kong can get access to it. I think this is one of the main difference between the US institutions and the local institutions in Hong Kong. So you actually get pretty good support from the university. So I think with that, I'm also a co-founder of a aging therapeutics company called Fountain Therapeutics. It is is located in the South San Francisco. So I work in the US time, and also I work in Hong Kong time, of course. So with that, that's me. So I think today what I would like to talk to you about is I use a few minutes to tell you what we do in my lab. So my lab interest is to try to understand the molecular regulation that regulates stem cell quiescence. And as it turns out, the disruption of quiescence will contribute to a depletion of stem cell pool, and particularly evident during aging. And our lab is very interested in understanding, how do we actually ameliorate aging, and of course, devise aging therapeutics. So I think it's no stranger to this group of people here. That we have different paths to aging reversal. So in my lab, we work on different aspects. We do have a setup of heterochronic parabiosis, where we basically look at the young and old mouse. It's a interconnected pair of mice that share the same blood circulation of factors. So we also work on different calorie restriction and exercise models. So today, what I want to show you with some of the data is regarding the metabolic reprogramming. So the model system that we use in the lab is these muscle stem cells within the skeletal muscle. And these are the little guy right here. The pink cells that you see in this cross section of the skeletal muscle. So they are basically dormant. They don't do much. And only when there's an injury, there's an extrinsic stimuli, they will wake up and participate into the regeneration process. So now, this actually serves as a very good model for chronological aging, for the reason that you decoupled the replication of the cells, or I should say the cell divisions from these stem cells will just basically stay there for the rest of the life of the animal. And we use mouse model for these kind of studies, of course. So, within these stem cells during aging, over the years, we understand that there are different kinds of problems. There's ROS, DNA damage, aggregation of damaged proteins, and also, some problems with mitochondrial dysfunction. So today, the story will be centered around the this particular aspect, the mitochondrial dysfunction. So to start with, I just want to show you some of the phenotype that you can see in, in some old mice, right? So if we were to take some of the young muscle, injure them, and or we take some of the very old, these geriatric TA, when I say geriatric, they are actually 32 months old. Like the end of life of a mouse is plus minus, I would say 36 months. So these are representing mice of very, very old age. And what you can see is that, the little round circles, these are the regenerated fibers. They don't look very well. They don't look very good in the very old mice. So if we were to look at the proteome of these stem cells within either the adult, the young adult, the old, or the geriatric, the very old. So purify the stem cells and subject that to mass spectrometry. What we can see is that, interestingly, the geriatric stem cells now possess a senescence phenotype, suggesting that the cells are actually doing bad. They're undergoing senescence, and they also lost the autophagy. So now, looking at these proteins, so these proteins that are downregulated in the geriatric stem cells, and of which actually almost 20% of these proteins are actually mitochondrial proteins. And if we were to do a gene set enrichment analysis on these proteins, quite many of them are arranged in the metabolism, respiratory, electron transport chain, right, suggesting that there's a impairment of mitochondrial respiration. So if we were to take these stem cells and put it into a seahorse machine, which measures the mitochondrial respiration of the stem cells, what you can see is that the young cells is in blue. They have pretty good levels of mitochondrial respiration, but the geriatric stem cells have a huge reduction of the mitochondrial respiration. So in the lab, over the years, we have been spending quite a bit of time understanding family proteins called the CPEB. So these are RNA proteins that will bind to the 3' UTR of a messenger RNA. And basically what it does is to help enhance or increase the translation. So, and if we were to take a look at the CPEB4, particularly one of the family members of the CPEB family, what we can see is that the CPEB4, which is right here, the levels is decreased in different tissues during aging, and particularly in the stem cells. These are the muscle stem cells. You can see that in young, you can see a good levels of the CPEB4. In the old, you don't see it anymore. And if we were to do a conditional knockout of this particular gene in the satellite cells or these muscle stem cells, what you can see is that, interestingly, the muscle stem cell mitochondrial respiration also decreased just very much like the old geriatric stem cells. And the idea here is that, somehow when you knock down or when you have a dysfunction of the CPEB4, some of the mitochondrial proteins are downregulated. So interestingly, if we were to maintain the loss of CPEB4 over time, the cells eventually senesce. So what we found is that, if you just simply knock out one gene, it will actually induce senescence. And this is the kind of proteomics work that we did to illustrate this point. So I mentioned before, the CPEB4 is a RNA binding protein. And when we were to work on a RNA IP experiment by sequencing, what we can appreciate is that the CPEB4 indeed actually bind to many of the mitochondrial proteins, and many of which actually are in the electron transport chain in different kind of complexes. And if you can see that CPEB4, the protein actually are co-localizing a good portion of them are colocalizing with the mitochondria. And if we were to do a CPB4 IP, I basically just try to look at the interaction partner subjected to mass spectrometry. We can also see that CPB4 actually binds to many different mitochondrial protein components. So now, take a look at the CPB4, the loss of CPB4 in muscle stem cells, and what we can find is that the function of the stem cells is impaired, which is illustrated in this diagram here. The muscle fiber regeneration is not as good. They are not able to proliferate as well. And if we were to restore the CPB4 level in the old cells and do a transplantation, what we were able to do is, the stem cells actually now regain the mitochondrial respiration, and we were able to engraft the stem cell in the muscle much better. And interestingly, this is also the last data slide that I want to show. If we take different cell lines of different human cell lines and basically induce senescence, right? Induce senescence, and this is the hydrogen peroxide induction. So you can see that the beta gal staining, they're pronounced in the induced model. And if we were to overexpress CPB4, it can actually suppress it, suggesting that indeed, having CPB4 can prevent ROS-induced senescence in human cells. So what I just told you in the last few minutes is that, we think CPB4 is something very important. It actually helps to maintain the translation of many of the mitochondrial genes, and thereby basically maintain a healthy mitochondria for normal stem cell function. And during the aging process, the geriatric mice lost levels of CPB4, and you also lose the mitochondrial function, impair stem cell function, and eventually get to the point that you get a senescence problem. So with that, basically, this is our beautiful campus, and this is the front entrance actually. So these are, as you go in here, that's the main building. So that's the kind of I would say science part that I tried to share. So I know there are only a few of us here. So I actually I don't know what is the best way to start, but maybe we can try to see if there's any questions first, and then we can get going from there.
And if anyone has questions, you can type them in the chat, or you can unmute yourself and just ask them.
Yes, Hitoshi, right?
Oh, yes. Thank you very much for a great talk. So I want to take this opportunity to ask you a question about your work. I think it's, yes, great work. I was a bit surprised that, if you overexpress the gene of your interest, you can just regenerate the aging muscles. But I'm not, I'm not familiar with muscle stem cells in general. So I want to ask you about how muscle stem cells are maintained over the aging status. Why you think it's so important to maintain the mitochondrial function during the aging? What happened to the mitochondria itself during the aging?
So I think that's a great question, Hitoshi. The long story short is I don't have a good answer for you. Okay, so but I'll try my best to postulate. Okay, so I think over the aging. Well, first of all, I would say these muscle stem cells. The interesting thing about them is, as I mentioned, they don't divide basically throughout the life the animal. I mean, unless you stress them, right? Meaning you injure the muscle. I mean, they basically just sit there for the rest of their lives. So, I mean, with that said, I mean, all we understand is, I mean, it is evident that the mitochondrial function is not as good in the old mice, and we, I mean, we have done, you know, different kinds of techniques to test that. I mean, like in the stem cells and actually in the muscle as well. I mean, the mitochondrial function is impaired. So I mean, but when we were to, you know, work on this project, I mean, I think the two parts actually just come together. I mean, the CPEB4 part, I mean, which we found that by surprise that it actually regulates all sorts of mitochondrial genes. And when we look at the old mice, we found that the protein levels goes down. So I don't have a very good answer for you, I have to say. I know, I mean, it's not exactly what you're looking for. So but I would say, I mean, the mitochondria, I mean, of course, in our view, what needs to be done is what's upstream from CPEB4, right? I mean, what regulates the function of the mitochondria, and why does it get dysregulated during aging?
Right. Yes. I also wonder about the upstream of this signaling, right? Because I mean, if I understood correctly, I think muscle stem cells very much quiescent, and it requires a certain maybe, you know, metabolic level, right, to maintain themselves.
That's right.
What I could understand was like, I mean, how these mitochondria itself are maintained. Do they divide? Even cells are quiescent?
So that's actually a great question. I have never thought about that. I don't know. I mean, I must say I don't know. I mean, even though I work on quiescence for quite a long time. So that's a great question. But I mean, like from our work, I mean, I think the mitochondrial function in the quiescent cells is rather intense. I mean, meaning they maintain a pretty good output. So I mean, I think they're quite functional, but whether they're divided, I'm not sure. Yeah. Mm-hmm.
Okay, thank you.
Yeah. Mm-hmm. So yeah, I think I mean, maybe we can talk a little bit about, I don't know, about aging. so I'm sure you're all, you know, in the area. So maybe we can, I mean, like, as you can see, right? I mean, like, our lab actually are very interested in understanding, I mean, how can we understand, you know, the process of aging, and of course, better yet, I mean, how can we perform some agent intervention, right? I mean, I think one of the question there would be, you know, when should we do agent intervention, right? Should we do it very old? Should we do it in the midlife, or should we do it very young, right? So when we do it very young, would that be a problem? I mean, is that does it consider as, I don't know, enhancement, right? I mean, like we then will live, you know, for a very long time. So would love to hear, you know, some of your views, if any, about, you know, when should we actually do, you know, some, I mean, if we have a good solution for agent intervention, in your view, what would be a good time to do this? So maybe I'll answer it myself. So I'm more of a proponent of doing it earlier. So I do think, I mean, if we can try to, I mean, first of all, I think exercise is one of those, right? I mean, we understand that exercise is a good intervention, right? I mean, exercise improve our function. I mean, generally speaking. So I think no one will say that we should not do exercise earlier, right? So I will be more on the side of, we should try to, if we know of a good intervention, we should try to do it a little earlier, if possible. Yeah. So maybe I would also say a few words about, I don't know, gaps and opportunities, right? I think we're all here in the area of healthy longevity, right? What would be the gaps? Actually, Alex has a question. Maybe we should answer that first. I was wondering why. So the question is whether the algorithm that we use to classify proteins, and what is your thought regarding age-related degradation in mitochondria and protein changes due to that? So first of all, we are using a mass spectrometry. The software that we're using is called Peaks. So it actually comes with the Broker timsTOF Pro machine. So it's a Broker proprietary software. So we use that to do a relative quantitation of the protein levels of our samples. So my thoughts regarding the age-related degradation in mitochondria. So I would, of course, from my short talk, what I would say is there is some translation issues. I think there's some issues with the replenishment of the mitochondrial proteins. So with that, I believe it has something to do with some of these RNA binding proteins. I think RNA binding proteins are something that not a lot of people have looked into in the process of aging. So in our group, we spend quite a bit of time trying to understand, if these proteins has the role in, regulating translation, the question there becomes, we understand that translation actually slows down during aging. Are these protein actually the levels of which these proteins are actually dysregulated in the old cells, right? So these are the kind of thoughts that we constantly chase after. So I hope I answer your question. Okay, Alex. Yeah, thank you. Yeah, so I was talking a little bit about gaps and opportunities, right? So I think in my opinion, the gaps definitely is we don't know a lot about aging, right? So and in my opinion, in the next little while, what we need to do is to collect more data, right? And in particular, if we have to collect data, these data should be, I hope, someone will actually do some longitudinal data, right? These, data that we can actually look at, people that, you chase the people for a number of years to get the longitudinal data, and so we can understand the changes. So of course, that presents from the gaps, we don't have the data to the opportunity. We can analyze these data and in a number of years. So what we really have to do, in my opinion, is how do we actually be smart about collecting data right now, right? So that's my view of how should we do or what are the gaps for now? Yeah. So I would say maybe one more question. Healthy longevity, right? That's what we're all doing here, right? So I guess healthy longevity, to some degree, suggests that we don't want to have a disease, right? I essentially, it's all about not having a disease, right? So I guess the question is, how do we do that? I mean, like, or what is the kind of a future thinking of, you know, the concept of healthy longevity? I mean, how do we actually define healthy longevity in a biological sense, right? I mean, like, what does it mean by, you know, having a cell that is healthy, right? So I think these are the kind of questions that I would love to understand and hear a little bit more over the you know, the summit, and also the future conferences and workshops that NAM is putting together. Okay, so I guess little bit quiet. I hope someone will enjoy the recording at least. But in any case, yeah, very happy to get a chance to share our work, and also have the opportunity to I mean, to think about, you know, this whole I mean, what does it mean by healthy longevity? I mean, at least that's the kind of view that I have. Yeah.
Thank you very much for presenting
thank you.
for us and answering our questions. And we encourage everyone to check out the other breakout sessions and the rest of the summit.
Great.
Thank you very much.
Yeah. So I guess I can log out also, right, Emma?
Yes.
Thank you.
I will go
stop
ahead
sharing first.
and I will end the meeting.
Okay. Thank you. You have a good day.
You too.